Hello, Dr. Esa. Welcome to the call. We're very much glad to have you around. Please, can you try testing your mic and perhaps share your screen? So kick off, sir.
Can you hear me clearly? Hello, can you hear me clearly? Hello, Daramala, can you hear me?
We can hear you, sir. So please try again now, sir. It's the screen. Can you try sharing your screen, sir? Your presentation. Yes, it's up now. Thank you very much. All right. Welcome back, everyone. We're glad to have everyone on this call once again. I trust I'm very visible. I'm very audible, rather. So earlier today we had Professor Malakroki, and it was a very wonderful session with loads of advice. I'm sure a lot of us picked something from that session. It was a very, very amazing and wonderful session. And this time around we have Dr. Temitope Faleye, who would be teaching us again this afternoon, depending on where we are anyway. So Dr. Faleye is one of the people who has really impacted us in IBRT, the team hosting this webinar. And we are very much glad to have him talk to us this afternoon. He's a faculty member of Ibadan Bacteriophage research team, and he has a PhD in biology from the University of Ibadan, Nigeria. He is currently a postdoctoral fellow at Arizona State University, where his research is focused on coupling case and wastewater-based surveillance with high-throughput sequencing strategies to understand dynamics of medically important viruses in human population. Currently, he has over 60 research articles, in reputable academic journals, and he has submitted over 1,000 sequences and raw reads to GenBack and SRA. So we'll be having Dr. Faleye speak to us on enteric viruses, high throughput sequencing, and genomic education for undergraduates. And we are very much excited to see him talk to us this afternoon. Welcome, Doctor. Everyone can just join me to welcome Doctor in the chat box as I hand over to Dr. Faleye to commence this presentation. Thank you very much. You can go ahead with the presentation now.
Can you hear me, please?
Yes, loud and clear.
Okay. Thank you very much for the feedback. I felt I wasn't something was wrong with my audio. Just one last time, please. Can you see my screen and can you hear me?
Yes, the
Please,
screen
can
is up.
someone confirm
And
that I'm sharing
yes,
my
the screen
screen
is up.
and I can
Yes,
be heard
the screen
clearly?
is up, and yes, you are very audible.
And okay, thank you very much.
Yes, thank you.
My name is Faleye Temitope. I'm a postdoctoral fellow at the Design Center for Environmental Health Engineering at Arizona State University. I'm grateful for the opportunity. A big thank you to the organizers. Today, I'll be talking about enteric viruses, high-throughput sequencing, and genomic education for undergraduates. This is my outline, and I'll be talking from the point of view of genomic epidemiology, but will be really focused on enteric viruses and high-throughput sequencing. So when we talk about genomic epidemiology, really, we're talking about the use of pathogen genomes to understand how infectious diseases spread through populations. And today, I'll be talking, when I say pathogen genomes are referring to viruses, I'll be really talking about how we use virus genomes to understand how these viruses are moving through populations. This is the layout of the conversation I'm going to be having today, or the methodology behind it. Really, what we do is we recover genomes from be it wastewater or feces or swabs. We recover virus particles from that source, and then we recover genomes from those virus particles, either by just extracting nucleic acid out of the virus, or in the event that you are looking for any particular virus of interest, you want to amplify either the whole genome of that virus or a portion of the genome of the virus, and then we sequence that. And when I say we sequence, what I mean is we read out the A, T, Gs, and Cs using whatever platform that is available. The data we get is then analyzed to make sense of how the virus of interest is moving through populations. Just so that we're on the same page, I'll be talking about enteric viruses and high-throughput sequencing, and I would like to clarify what I mean by both before we dive into the conversation. When I refer to enteric viruses, I'm referring to viruses that loosely, at some point in their replication cycle or transmission cycle, get to replicate in cells of the gastrointestinal tract or cells that are associated with the gastrointestinal tract. In my talk today, I'll be talking largely about naked viruses, not envelope viruses, because I know the way I've defined enteric viruses, some envelope viruses would fall into that category. But my talk today will be focused on naked viruses. And by naked and envelope viruses, what I mean is that most viruses of medical importance, you can actually group into two morphology. There are those that have a lipid bilayer surrounding the virus particle or the nucleocapsid. Those we refer to as envelope viruses, and those that don't have that lipid bilayer, which means they just have a protein coat, and then within that protein coat, you have the genome. Those are referred to as naked viruses. So the two viruses up here are the classic example of the kind of viruses I'll be talking about. And these viruses are of immense medical importance because they are associated with a range of clinical manifestations. One of those that best known is poliovirus. Poliovirus falls into this category, causing clinical manifestations in the CNS. Other viruses like adenovirus, which cause gastroenteritis and diarrhea, are some of the viruses in this group. And it's been estimated that these viruses are responsible for over 700 million infections annually globally, and quite a significant amount of morbidity and mortality. So when I talk about high throughput sequencing, I want to draw a line between that and Sanger sequencing, at least just to make it clear in our minds that Sanger sequencing, the basic difference between the two is the amount of sequencing you are doing at any point in time. With Sanger, you have to sequence amplicons one after the other. With high throughput sequencing strategies, you are sequencing millions of fragments simultaneously per run. And the logic is this: whether you are working directly with genome that you have extracted from the virus particle, or you've amplified a segment of the genome, you take whatever amplicon you have, you break it into fragments, add adapters to the ends, and then make libraries with that. The library then gets sequenced and the data is captured. And because you started out fragmenting that genome, to be able to get your sequence back or your genome back, you have to now assemble those fragments to make contigs. This data that comes out of your sequencer in short fragments that you end up assembling, we refer to them as raw reads. And that is largely, the raw materials from which all the other data that you'll be generating will be coming from. And this slide describes what I'm going to be talking about today, the model after which all my talk today will be patterned. So when we get these raw reads, most of the time, because of the amount of sequencing we're doing, you end up sequencing other things that are really not your virus of interest. And then you de novo assemble, recover contigs for the viruses you're interested in, and then you map those contigs back to your raw reads to actually extract the virus you're interested in. And usually when you do this with enteric viruses or with other wastewater or fecal samples, most of the time your virus of interest is about 50% or at times less of what you sequence. You have this remaining 50% that might not be directly of interest. What at times would happen is people submit all the raw reads to the SRA, and then we'll talk about their contigs and all. So, but what we've been trying to do is we map these reads, submit the map reads, recover what is left, and then try to farm that for bacteriophage genomes. And those genomes we find, we then look for those that are novel, that are not represented in any database, and then use that to train our undergraduates, to educate them on what genomes look like, how genomes are structured, how do you find open reading frames in genomes, how do you compare the architecture of different genomes, then how do you make phylogenetic trees, what is the logic behind it, how do you interpret trees, what do trees mean, and then in instances where you are finding it hard to understand what the function of a protein is, because at times it could be novel, you want to predict the structure and then see where the active sites are, what are the amino acid residues that are involved in particular activity, what are the binding sites for DNA binding proteins, those kind of things. We want to have students move from learning things in theory to actually handling these things in silico and beginning to translate and apply the knowledge, the theoretical knowledge that they have gained. We also try to ensure that all this knowledge, gets moved forward. The students go on and present at conferences, they write manuscripts and publish. And then one thing we've also been trying to do is we've realized that because a lot of this is in silico work, it makes a difference when the students have something to hold on to. So what we've tried to incorporate into this is when we predict structures we pick a protein at random, then we 3D print that protein, make it into a kitchen, export the 3D structure out of Chimera X or Chimera, put it in something like Tinkercad, add a keychain to it, and then 3D print it. And there's a lot of aha moments when students hold the protein structure in their hands and they are going over the details they've pulled out of literature, pulled out of analysis. It's a different feel that doesn't come when there's that tactile part of it has not gotten into the conversation. So I'll try to, in the course of my talk, I'll be talking about this angle and I'll be talking about this angle simultaneously, and I'll do that for 2 projects. I will talk about 2 projects I'm involved in, one with wastewater in Arizona, the other with non-human primates in Nigeria, and I will show you how we're doing genomic epidemiology on both sides and how we're getting data and training undergraduates on what genomes are and how to think about them. So first, let me talk about the wastewater-based genomic epidemiology work. So the first question that would come to you is, why would you want to look for viruses in wastewater? And that is largely because usually less than 10% of infections end up showing clinical manifestations. So most of the time, say out of 10 people that get infected, 1 person goes to the hospital, and that is what case-based surveillance captures. The remaining 90%, despite the fact that they're shedding virus in large quantities, don't end up showing up in hospitals, but they all shed virus in feces or urine or whatever, goes to the treatment plant. So technically, what shows up in hospitals is just the tip of the outbreak iceberg. And what you are able to do with wastewater-based genomic epidemiology is you have a cost-effective strategy, both in money, time, and manpower, to screen thousands to hundreds of thousands of individuals per sampling event. Like in the study I'll be talking about, we're collecting samples from about 700,000 people. So in one shot, you can look at virus present in a population of 700,000 people, and it's non-invasive. You're not going to knock on anybody's door. You're not, it also gives you opportunity for a One Health approach, meaning that any pathogen or any virus in animals in that population that gets into the wastewater system, you can find that too. Furthermore, it gives us an early warning system, because usually before you start seeing clinical manifestations in hospitals, at times you would have had an outbreak going on for weeks, for months, for days. Wastewater-based epidemiology gives you that lead time to see things before things really get bad, and allows you to intervene early. If well designed, it can also, provide information on where whatever is circulating in your population has come from, where's the source of the introduction, and it can also help you determine whether whatever arsenals you have with respect to vaccines or antiviral agents, can they work for your pathogen of interest. And if you need to start thinking about new vaccines, thinking about new antivirals, it can, give you a lot of lead time to get that done. So just to be on the same page, this is the general workflow of the conversation I'll be having with respect to wastewater-based epidemiology. We collect wastewater, we concentrate, then we recover nucleic acid from it. Usually, we amplify the portion of the genome we're interested in. We do Illumina sequencing, and then we analyze the data to make sense of it. In this particular study, let me give you a little bit of backdrop to what led to this study, but it's already publicly available in medArchive and currently under review. At the start of the pandemic, a lot of research has turned to wastewater-based epidemiology. Wastewater-based epidemiology has been around for a long time, and environmental surveillance is going on for polio, and that has been on for decades, and quite a number of other viruses. But at the onset of the pandemic, people were looking in wastewater to see if they could glean information from wastewater about what is happening with SARS-CoV-2. And we found that it seemed there were two schools of thought. So because virus concentration in wastewater, I mean viruses of medical importance, because viruses are obligate intracellular parasites, and viruses that infect humans usually don't replicate in wastewater. Viruses of bacteria, bacteriophages on the other hand, will replicate in wastewater because bacteria would multiply in wastewater and their viruses can infect them. And so they are usually the underdogs in that environment. But at the same time, they are very dilute. So you want to take a large volume, take all the virus that is in there, and then screen for it. And how one of the ways you do that, one very efficient way that has actually gained prominence with SARS-CoV-2 wastewater epidemiology is you use two filters. You have one filter that is about 10 times larger than the size of your virus. You pour your sample through that filter. That way, bacteria and undissolved fecal matter, all of that gets trapped by that filter. And that is the filter here for 15 nanometers. And then you now have another filter that is about 10 times smaller than the diameter of your virus. So this second filter traps your virus in it, and then all the water gets out. Then you can recover what is in there and get whatever virus is in suspension here that was not trapped here. But there were also people that were looking at what was trapped in this 450 nanometer filter, and we're finding virus in it too. And some people don't even use two filters. First, they will just centrifuge, get the heavy particles in a pellet, look for virus in that pellet. They were finding virus in there, and some also finding virus in here. So there were papers that came out that were looking at both fractions and were asking, can we use what is in this fraction to estimate what is in that fraction? And they would do real-time PCR. And by that, what I mean is they would measure the concentration of virus here and measure the concentration of virus here and try to use one to predict the other. But as we watched the field evolve, we noticed nobody was asking the other side of that question. While you can measure virus concentration, what about virus diversity? Are they one and the same thing? Because we know from basic virology that they are not the same thing. So what we try to do in this study is, rather than use virus concentration in one to look at the other, we're asking the diversity of viruses trapped here, are they the same as diversity of viruses trapped here? And can we use one to estimate the other? And to do that, we took 3 different viruses that have naked capsid, and enteric viruses are very stable in the environment, meaning that the genomes will be preserved well so that we can actually do long-range PCR and get a sense of the region of the genome that is important, that is under heavy immunological pressure, to get a sense of what is happening in the population. So we looked at enteric viruses which have over 300 distinct serological types, and poliovirus is a member of this group. They are of immense medical importance, associated with a range of clinical manifestations. We looked at adenovirus 41, which is associated with diarrhea in children largely below the age of 2, and recently has been associated with hepatitis of unknown origin. And we also looked at canine picornavirus, a virus that little is known about, seems to be a virus of animals, but I'll shed more light on it as I talk. And this is what we found. So let me give you a sense of what this is. This is a picture of the filter I just showed you. Everything that is in Phage, is what passed through the 450 nanometer filter and was in the filtrate. That is what is on this side is what we have here in Phage. And whatever was trapped in the filter is what we have in blue. And just looking at the image, you can see that for enterovirus, for canine picornavirus, and for adenovirus 41, you really can't use what was trapped in the filter with respect to diversity to predict what is in the filtrate. At times you have an overlap, but generally it seems like it's something that cannot, that it's I wouldn't say it's unrealistic, but we need to start looking at the samples differently and not saying, oh, we use one fraction to predict the other. The two of them are holding different things, and it seems that best practice would be to bring both together to, understand what is really in the population at any point in time. But beyond this, we're generating data about regions of the genome that was under heavy immunological pressure. And then we asked a couple of questions of the data we generated. So, okay, can we identify virus circulation in a population that is not documented by case-based surveillance? And I'll show you how we did this with enterovirus Cs. So why are enterovirus Cs important? Well, poliovirus is a member of enterovirus species C, and the world is on the verge of eradicating polio. There are two vaccines, the IPV and the OPV. IPV is a IPV is an inactivated polio vaccine. It's not a live vaccine. It is given as shots, but OPV, oral polio vaccine, is a live attenuated vaccine. It is given as drops in the mouth. And the beautiful thing about OPV is that it generates beautiful both humoral and mucosal immunity. IPV, on the other hand, generates very robust humoral immune response, not that good mucosal immunity. So when you have an outbreak and you want to interrupt transmission, usually you want to administer oral polio vaccine. The downside of oral polio vaccine is that it tends to revert to virulence. Like I said, it's an attenuated virus, but after rounds of replication, it tends to regain pathogenicity because it loses certain gatekeeper mutations. However, being a member of enterovirus species C, what has also been shown, in fact, when you define virus species, the classic definition of species is that members of the same species, when they co-infect a cell, can share parts, can recombine, and still produce infectious particles. So when poliovirus infects a cell that is co-infected with some of these non-polio enterovirus species, they tend to recombine and take non-structural region from these non-polio species and also regain transmissibility and pathogenicity. And there are several parts of the world now where we have cases of vaccine-derived poliovirus, circulating vaccine-derived poliovirus going on. However, there had been a narrative that that the reason you have circulating vaccine-derived poliovirus in sub-Saharan Africa and some parts of Asia is because you have an overabundance of enterovirus species. But you don't have that happening in, allow me to say, developed nations. There was a paper that came out in 2021 where Bro and others went to GenBank and downloaded enterovirus data and stratified them by the part of the world where the different species were recovered. And you can see from the data that it seems as if species Cs are not in the US. But when we look in wastewater in the US, we see that over 50% of the variants we are recovering are actually species Cs. And when we now take those sequences and go to GenBank and do a blast search, we realize that they are super divergent from whatever is present in GenBank. In fact, poliovirus is so well clocked that when you pick two poliovirus vaccine variants, you can tell how long they've been circulating because their structural region evolves by about 1% every year. So if you go by that clock, finding a virus circulating in the population that is about 20% divergent from whatever is closest to it suggests that there are viruses that have been circulating for around 20 years without being sampled in this population. And of course, if it's either they are not being sampled or they are being sampled, not made publicly available, sequence not made publicly available. And if that is the case, then it makes sense that Brown and others would not find the data in GenBank when actually the virus is actually circulating in the population. Earlier this year, around June 2022, there was a case of poliomyelitis in New York in a man that is greater than 20 years old. And subsequent environmental surveillance showed that there was actually poliovirus, circulating vaccine-derived poliovirus to circulation in surrounding counties. The interesting part, however, is that when they looked at the capsid region of that virus, it seemed to have been circulating for about a year. And when they looked at the P3 genomic region, they see that it is a recombinant virus. The green here is from the virus of interest that was isolated in New York. The interesting part, however, is that if you take that P3 region and you do a blast search, besides the other, because this particular virus has been found in the US, UK, Israel, you do a blast search, the most similar things to it in GenBank, beside viruses that are associated with the outbreak, are about 80-something% similar to it, suggesting that the non-polio species C that are combined with that virus is virus that is circulating in a population that has not been sampled for possibly over a decade. Of course, the way you would clock a non-structural region is different from because they are under different pressures, but it further confirms what we had found, that actually there's virus circulating in parts of the world that have not been sampled for decades. That is what one important thing that this outbreak of vaccine-derived poliovirus in developed nations is showing us. There are pockets of enterovirus species C circulating in parts of the world, possibly not sub-Saharan Africa or Southeast Asia, or Asia, allow me to say that, because there have been a lot of VDPVs there, and technically those VDPVs are like poliovirus sampling non-polio species C sequences circulating in those populations. And the fact that we can't see anything that looks like that suggests to us that EVCs are circulating in these populations. They're not showing up in the clinic. And one way to actually look for these things is, as we have shown here, to use wastewater-based genomic epidemiology. And the beauty of this is that it allows us to see things to come ahead of time so that we can make informed decisions based on the insight we generate. In fact, in the paper we published last year, we had mentioned that with what we were finding in wastewater, if poliovirus gets into these populations, because we were finding evidence of co-infection, recombination, EVCs, that imagine that CDVPs can actually emerge in these populations. And like a year after, we saw that happening. So this alludes to the power of wastewater-based epidemiology. It can help you really see things ahead of time and actually in an early warning capacity and make informed decisions. The next thing I'm going to talk about is what we have learned looking at adenovirus 41. And like I previously mentioned, it had recently been in the news. Over time, we had known adenovirus F41 as an agent of diarrhea in children below the age of 2, and sometimes could cause mortality in people with immunodeficiencies. But recently, they were associated with cases of hepatitis of unknown origin. So we went back into our samples and said, okay, let's look for these viruses. And what we did was we amplified whole genomes in overlapping fragments. And because they are DNA viruses, they are not they don't evolve as fast as RNA viruses. So we also have to focus on the capsid region and portion of the capsid that are evolving very fast. And here we focused on the hexon protein coding sequence. And what we did was, because they're not evolving very fast, when you do de novo assembly, you just get one config. So we use that config to go to GenBank and look for the most similar config in GenBank or genome in GenBank, and then use that as template for variant profiling. And what we found was we had 7 variant profiles. But really, first, we could tell that you can't use one fraction of wastewater, be it what was trapped in the filter, to estimate what is in the filtrate. You had different variant profiles in most of them, except for January 2020. But to now translate these variant profiles to what had been found in the children with hepatitis of unknown origin in 2022, we had to go back to GenBank, download whole genomes, extract whole genomes of adenovirus F41. I think at that time we had maybe 59, extracted the hexon gene from them, and then use those exon genes to now search GenBank for more complete exon genes that were not in whole genomes. And we picked the top 100, used it in phylogenetic analysis, and then we now did pairwise identity to get a sense of the amount of each cluster within these trees. And we learned that each cluster has less than 0.4% divergence within it. We then look for amino acid signatures of each of those lineages, and then use that as some form of dictionary to interpret the exon lineages that we had found in wastewater. And we learned, and what that dictionary translates to us is that lineage 1 is the same as variant profile 1 or exon profile 1, lineage 2 is the same as 5, lineage 3 has profile 3 in it, and exon profile 7 is a combination of lineage 5 and 6 in it. We couldn't find exon profile 2, 4, and 6 in the data that was in GenBank as at that time, and that might suggest to us that there are actually lineages out there that have not possibly gotten into clinics, talk less of being sequenced and made publicly available. So on that data now, knowing these amino acid signatures of the exon hypervariable region of exon gene of this viruses, we then went back to data that had been published about children with hepatitis of unknown origin and layered that upon it, and we could show that these viruses from the US, children with hepatitis of unknown origin in the US, actually belongs to lineage 1, lineage 2, and lineage 6. And what we show here is that lineage 1, lineage 2, and lineage 6 have been circulating in the US from way back as 2019. This further alludes to the power of this approach, wastewater-based genomic epidemiology, to help you to go back in time and look for signals you might have missed, and also help you, focus on assays that help to now, going forward, begin to look for things that might not show up in clinics, and guide us on how to look for those things. The other lesson we learned is how to find emerging viruses. And here I define emerging viruses as a virus that's been found in one population but not in another, and is now new in a new population. To explain that, I'll be talking about canine picornavirus and what we found. Canine picornavirus were first documented in 2012. And between 2012 and 2019, there were 6 genomes in GenBank describing canine picornaviruses, and they were found in dogs and red foxes in Hong Kong, China, United Arab Emirates, and Australia. And they were from samples that were collected between 2008 and 2019. So it looked like something that was, in Asia and Australia, until looking for enteroviruses, our assays started pulling out, it was just serendipity that our assays were pulling out this virus in wastewater. We first found it in, I think, Illinois, Kentucky, and then we found it in Arizona, and we now had to go back and actually design assays for it and try to pull it out of wastewater. And what we found is that based on the data, our phylogenetic analysis and POS identity, there seem to be 7 genotypes of this virus that we've documented till date. And genotypes 5, 6, and 7 for now seem to only have been described in the US. Genotypes 1 and 2 have been found in Asia and the United States. Genotypes 3 only in United Arab Emirates, 4 only in red foxes and in Australia. And it's important I mention that as of now, we've only found this virus in wastewater in the US. We don't know where it is circulating. We know it is circulating. We don't know what the host is. The possibility exists that it's infecting canines, as has been found in other parts of the world. And the US has over 70 million dogs, and most of them are pets living in people's houses. Of course, there are other canines that could be hosts, but it's interesting that you could have a virus circulating, possibly circulating in canines that have not been sampled. And our phylogenetic analysis suggests that these things have been here because we see divergence going upwards of 15%. And our estimation of the evolutionary rate of the virus suggests that these viruses are already exotic in the US and might have been here for over 15, close to 20 years, circulating, yet there have been no description of their isolation anywhere in the US, despite the fact that they're circulating. This alludes to the power of wastewater-based genomic epidemiology to help you actually see what viruses are present in populations and how they are behaving. In fact, we did this over 2 years, 2019, 2020, 2021, and the difference between those 2 years is before the pandemic and first year of the pandemic. And the way the virus behaved in wastewater was exactly the way enteroviruses behaved in wastewater. We had like a 4-fold drop in diversity. And that makes us begin to think about, because we've not seen data describing viruses of animals have that shift in diversity during the pandemic, because of course we know there were non-pharmaceutical interventions like lockdowns and all that resulted in that. We don't know if that impacted virus diversity in pets. We don't know if it in fact impacted virus diversity in animals, but the only information that we have from the pandemic largely is about how it impacted virus diversity in humans, and this perfectly mimics virus diversity in humans. So it's getting us thinking. We're not saying the virus is infecting humans. We don't even know what is infecting yet. But it's an interesting exercise to actually see that there are things going on in populations that are not showing up in clinics yet. And because prior to this study, nobody had tried to genotype the virus. So we first had to genotype it before we could now look at how it was behaving in the filter and in the filtrate. And we saw that you also really couldn't use one partition to predict the other. So now, in addition to this, like I previously mentioned, we have a scenario where in wastewater, phages are the predominant virus population in wastewater. And despite the fact that we are amplifying, using PCR to first amplify our viruses that we are interested in, only about 50% of the raw reads we generate actually map to these 3 viruses I've talked about. The remaining 50%, majority of them are phage genomes and other things. So what we then tried to do was to not just take all the phage genomes that were present, look for those that seem to be unique, seem not to have been described anywhere before. So we take the genome and use it as educational tools for our undergraduates. And here I'm going to talk about one of those genomes. This is one of the genomes we found. We had more than 100 times mean coverage, meaning that each nucleotide sequence each nucleotide position was sequenced more than 100 times. And this is despite the fact that we did amplicon-based sequencing for viruses of medical importance. So we're not looking for this. We still have this depth of coverage for the virus. And a blast search showed that most likely it's a microvirus, but you can see query cover is just about 10%, meaning that it's really not similar to anything that is present in GenBank. So these are the two undergraduates that led this work, presenting the work at the seminar. And this is the poster. And I'm going to just walk through it and help us get a sense of what they did. And I have to acknowledge that these are very talented young people, very brilliant minds. And what did they do? They took the genome, did a blast search, a mega blast, didn't look like anything, that they converted to a blast end search and saw that, okay, they think this is actually a microvirus, and then went ahead and did genome annotation using two different softwares to predict the open reading frames that are present in the genome. And then with the BLAST-P search, they could annotate most of the genomes and determine what their functions were. They extracted the microvirus capsid protein and then used that to download more microvirus capsid protein from GenBank, did phylogenetic analysis to classify the virus, and okay, this is what it is. However, there was one protein that we could not figure out what function it had, because when you do a blast search, what it was most similar to was about 30% identical, and even that was an hypothetical protein, so they had to figure out what it was. So using HHFRED, they were able to see that there are signatures that this has a helix-turn helix motif and seems to be a DNA binding protein. Then they went ahead and predicted the structure of the protein using collab fold, which implements AlphaFold 2 with MMSec, and this is the predicted structure of the protein. They then did use dRNA prep to predict nucleic acid binding residues, and we found a cluster of those residues on the protein, and then downloaded the protein 3D structure, converted it, put it into Tinkercad, and then put a ring on it and tried to make it into a keychain and 3D printed it. And it's one thing to do all these in silico analysis and predict protein structure and all of that, but then having to hold the protein, there were a lot of aha moments with the students. Oh, okay, so this is what it looks like. Oh, look at the curvature. Look, of course, we know this is our best guesstimate, but it was enlightening and it was a different feel, and it's like it brought the science to life. And if you're interested in reading about it, it's already published, and those are the two students that led the work. What we try to show here is that while now there are a lot of groups that are doing genomic epidemiology with SARS-CoV-2 and MinION and all this sequencing capacity in different parts of the world, all these laboratories are generating data. And all this data, while a good proportion of it is going into genomic epidemiology virus of interest, there's a lot of data being generated that might not be directly, going into that workflow. This data is valuable and can be a very essential resource that can help educate undergraduates globally on what genomes are, how genomes look, how to make sense of it, how to extract information from genomes. And currently, the undergraduates in the group and research associates are working on another genome. A megablast says it doesn't look like anything. The BLAST-10 shows that you have about 40% query cover. It looks like a phage that infects rhizobium. This is the most similar thing in GenBank. They've done structure, they've done annotation. In fact, in this case, we have 3 proteins whose function we really don't know. This doesn't even look like anything in GenBank, excuse me. And we also have an endolysine, and endolysins are bacterial proteins that chop peptidoglycan and are finding application nowadays as alternatives. With the advent of the predominance of antibiotic-resistant bacteria, there's a lot of shift towards using phages as in clinics or in controlling bacterial infections, and endolysin are actually finding center stage in a lot of applications. So predicting the structure of these endolysins, trying to figure out what it cleaves, how it works. We know that it belongs to the peptidase family M15. It might require a ligand, and using both AlphaFold 2 and ITASA, trying to get to better understand. And this is work that is still ongoing. Hopefully, maybe before the end of the year, this will be completed and should be published. And it's really an exciting opportunity for undergraduates to actually, go beyond. And like I said, we try not to just annotate all the genomes, all the phages we find. We look for those that are novel so that it can really be a learning experience for the undergraduates. And we are not just doing this, like I said, with wastewater. I'm now going to talk about using exactly the same model for work that is going on in Nigeria with non-human primates. So in Nigeria, annually, you have more than 5,000 cases of acute flaccid paralysis or suspected acute flaccid paralysis that end up going into AFP surveillance systems. And of these samples, you tend to have less than 10% have virus isolated in culture on RD and L20B cell lines, which are the cell lines that are used in the program for isolating polio and quite a number of non-polio enteroviruses. If you use direct detection, that number goes to about 20 or a bit more than that percent of the samples. But a very minuscule proportion of those samples ultimately have polio in them. But around 2015, 2016, we started seeing cases of viruses that have been, enteroviruses that have been described in non-human primates in other parts of the world in children with AFP in Nigeria. And that really got us thinking. And we said, okay, let's look at, can we find enteroviruses in non-human primates in Nigeria? And we got samples from Jos and from Cross River State. And interestingly, in the sample from Jos, we found an enterovirus A76 in a chimpanzee that was in a zoo. That animal had been born in the zoo, had been raised in the zoo all its life. And we found an enterovirus A76 in it that clustered with variants from Southeast Asia. From Asia, that was insightful for us because we also had EVA76 in humans in AFE, in humans in Nigeria, and those viruses were not anything close to what we found in that animal. That suggested or confirmed, findings we had that there was virus coming in from Asia into the country and all. But with the virus the virus we recovered from the animal from Cross River was Ecovirus 11, and the Ecovirus 11 we recovered here belonged to a cluster that was found in wastewater in 2012 and in 2014. And a virus in that cluster was also found in the Netherlands in clinical presentation. And in fact, we sequenced the whole genome of this particular Ecovirus 11, and we showed that the non-structural regions were most similar to that of viruses circulating in humans. And these two studies made it clear to us that indeed there was virus going from humans into non-human primates in Nigeria. And of course, what led us to this was that we're finding viruses in humans that had been shown in non-human primates. And that got us thinking about what really was in these non-human primates. So we decided to take a metagenomic approach, meaning we take feces from these non-human primates. And this is work that, O.M. George was the really driver driving this work. And it was done in collaboration with Gilles Group in Belgium. And what we did was take the samples, recover virus nucleic acid, but before that, you have to clarify, suspend the fecal matter, clarify it, extract, destroy every nucleic acid that is in there that is not of viral origin or not within the capsid. Then after that, you now extract nucleic acid from whatever is trapped in the capsid, and then do Illumina sequencing and try to analyze that and figure out what is in there. And from the first animal that would analyzed that way, that we processed, we found a whole range of brand new viruses that belong to well-established DNA and RNA virus families, but never been described before. In some cases, we found brand new species, we found brand new strains based on ICTV criteria, we found brand new serotypes. And down here, I try to focus on this enterovirus J that we have up here. That was the first time that serotype was being found anywhere in the world. And I went back about 2 days ago and still picked the genome and did a blast search, and you can still see that despite the fact that this virus is from 2016, till date, you still cannot find anything in GenBank. This is it here, the first one, that really looks like it's in the VP1 and VP3 region. We've gone ahead beyond that point to sequence more genomes, and we found more EVJs and more of many other viruses that we feel might be of medical importance because members of those same genera are found in humans. For now, we found that there are 2 enterovirus J serological types in Nigeria that seem not to have been found anywhere else in the world. And we say that because, classically, enteroviruses were defined as serological types, and that means that if you infect with this virus, the immune response it elicits, if it can prevent infection of an animal or a cell by a different virus, we consider those viruses as belonging to the same serological type. However, if the immune response elicited in response to one virus does not protect against another, then we consider them as different serological types. And with molecular epidemiology, it became clear that you can define serological types by diversity in the VP1 region or the capsid region, and that members of the same serological type are less than 25% divergent. And so when you use that criteria now, and so what we did was we took this EVJ, did a blaster, picked the top 20 hits, and added the new sequences were generated, did phylogenetic analysis, and then did pairwise identity. And what you see automatically, our cutoff here is 25%. So all these red bars refer to or show you sequences that are within 25% divergence of each other. And you can see it correlates well with the topology of the tree and with strong bootstrap support. So we know that in these two parts of the country where we've been sampling NHPs, there are two distinct enteroviral species, J serological types, that are circulating in those populations. And we can see that there are other types too that are circulating in Cameroon. And we've been getting a lot of this in mandrills. So what, as I've been saying, we try to do here also is we took the sequences that are, the raw reads that map to genomes that seem to be are from viruses that have been found in humans that might have medical importance. And then, of course, it's a metagenomic study. We had a lot of phage genomes, and then we tried to look for novel phages in these two. And good for us, we have a very strong C phages team who are also the organizers of this webinar. They've been doing beautiful work annotating large bacteriophage genomes, and so they just pulled all their expertise into annotating these genomes and finding beautiful things. First is, like I said, we don't try to annotate all the phage genomes that are in there. We just pick things that are novel so that the students are actually contributing to science, not just going over the motions. And you can see that the 5 genomes that we've selected here, there's barely anything in GenBank that looks like them. You can see query cover 2%, query cover 3%, and these genomes have been annotated. The open reading frames in them have been predicted. We already know the predicted life cycle of a good number of these viruses, and we're trying to better understand some of these hypothetical proteins, what they are, predict function for quite a number of them, because really there isn't really anything in GenBank that looks like them. And we're applying the whole repertoire, we're doing the whole thing, phylogenetic analysis, protein structure prediction. We intend to also, they're already presenting posters, writing manuscripts, and we have not decided on which of the proteins will be 3D printing, so that at least, to give that feel for not just a purely in silico algorithm. The lessons we've learned here is that there are novel viruses everywhere, not just, at least for me, one thing is revealing to me that there are novel, there's, maybe it's my bias or maybe it's the way I interpret things. There seems to be this perception that novel viruses are in sub-Saharan Africa and Southeast Asia. And indeed, it's correct that there are novel viruses there, but I think there are novel viruses everywhere. And if particularly we're looking from a zoonotic point of view, I think our focus shouldn't just be, you want to look in forested parts of Sub-Saharan Africa and Asia. I've just shown in the previous part of this talk that there are quite novel viruses in the US too that are circulating, possibly in canines, that you have over 70 million of them living as pets in people's houses. So we need to have this balanced view, and the approach we are using shows us that there are novel viruses everywhere, in NHPs, in Nigeria, in every part of the world. And all these studies doing genomic epidemiology are generating data that might not be directly, translated into clinical care or understanding viruses that cause clinical manifestations in humans. But this data is still valuable for undergraduate genomic education, and I would implore us, as genomic sequencing capacity is becoming readily available all over the world, as many groups as have the capacity to begin to pull undergraduates in and train, and because where we are going, we need as many hands as we can get. We need people that can actually take this data and make sense of it and develop new tools and develop new capacities. So far I've shown you 2 projects that I've used this model that I've described here. We've used it with wastewater-based genomic epidemiology. We've used it with fecal samples from NHPs to understand what viruses are out there and how they are behaving in populations. We are doing the same thing with fecal samples from humans. Like I've already mentioned, that you have a lot of acute flaccid paralysis cases in different parts of the world for which the pathogens responsible are not being accounted for. We're trying to take a stab at this, and we're finding so many interesting viruses in these cases, and quite a number of them are quite fascinating. You're finding lineages that have never been described before, sublineages that have never been described before, lineages that seem to be confined to certain geographical parts of the world. And it's an exciting time. A lot of this data is still unpublished. It's analysis that is ongoing, and it's been done by an excellent group of virologists, both PhDs, graduate students and the Phage's team in Ibadan, Nigeria. And in conclusion, I would like to just go over the lessons I think I've tried to communicate in the course of this talk. That is, with genomic epidemiology, be it with wastewater or with samples from clinical cases or from animals, can enable us see things to come ahead of time in an early warning capacity and help us make informed decisions based on the insight that they generate. They can also give us the capacity to look back in time in cases where we have archive samples to see signals we might have missed and guide us on how to go forward in looking at how viruses are behaving in populations. They can also give us information about viruses that are in different locations but now finding their way into new geographical locations even before these things start showing up in clinics. This technology gives us the capacity to do that and also to find novel viruses and provides a lot of data that is valuable for undergraduate genomic education. And I would implore as many systems that have these capacities to pull in undergraduates I don't think there's a substitute for that those aha moments you see in the eyes of undergraduates when the pride and the joy and the excitement in their eyes when they do this, and they even win awards, they win best prizes, they get recognition for the work they are doing, not just theoretical but actually contributing to global science. And of course, I'm sure everybody knows that all the work I've presented here is work that has been done by a village, and I've been privileged to have spectacular mentors, have spectacular collaborators, working with me, and I feel particularly privileged. All the work I've talked about with wastewater is work that is supported by the LMNIH grant given to Professor Matthew Scott of Haldane and Avin Vasani at Arizona State University. And in that team, I'm privileged to be working with people like Erin, Sangate, Devin, Jill, Ibrana, Nicole, Ainsley, exceptional undergraduates and graduate students and techs. And the NHP work is work largely led by Dr. Adeniji and Dr. Dehumi, and really driven by O.M. George. And O.M. George is one exceptional field and molecular virologist that I think people should really look out for. And a big thank you to Dr. Sheyi, Dr. Noja, and the entire team in Ibadan that is, going over this AFP data and the NHP data and doing all the analysis. And a big thank you to Professor Ademowo, the lead of the Phage's program in Institute for Advanced Medical Research and Training. He's an excellent mentor. I've been privileged to have the opportunity to work with him and the faculty, the Phage's faculty, and the exceptional undergraduate students in Ibadan that are doing all the analysis with Phage data. A big thank you to everybody. Some of the references I've cited. Thank you very much for listening. I'm happy to take questions.
Thank you very much, sir. It's really been a wonderful and enlightening session. We really appreciate
Thank
this
you
session,
very much.
sir. So we have a hand up. Olayinka, please go ahead if you have a question. So you kindly drop your questions in the chat box, and if you have questions also, you can raise up your hand and we'll call on. So Olayinka, please go ahead.
Okay, thank you very much, the moderator. I'm so proud to have you, Dr. Faleye, for the lucid explanation of everything you're taking us through and for the quality of the information you've passed across. So I've learned a great deal, but I see this thing that you've shown us that the wastewater molecular epidemiology gives a bigger picture of the tip of the iceberg of what you actually feel at the clinical level. Am I audible, please?
Yes, we can hear you clearly, sir.
So, but could it be that the almost 90% of the cases that we don't feel at the clinical end, can we start to ask another question? That could there be a reason for why we have this barrier of the diversity that we have below the ladder, not coming up into the human population or into the animal population that we can feel? What could be the reasons, or what could be the barriers that we find a number of all these pathogens a number of all these uh, bacteriophages, viruses, that, well, we can only hold them at the non-infectious side of the epidemiology and not already in the chain of transmission. So can, can a field look into that in the nearest future? Do you feel that something like that could look into why we have so much down the ladder and too few having clinical importance? The second question is just about clocking, and it struck me when you actually said the non-structural um, part of the most genomes of the peculiar, or mostly non-structural areas of viruses are clocked differently. I just want to know the science why this is happening. Thank you very much.
Thank you very much, sir, for your kind words and beautiful question. As regards what informs clinical manifestation, so this is the way I think about it. It's like you have intelligence agencies all over the world, and what they do is, in some cases, they allow me to use the phrase, they go into the dark web or go into human society to try to flag things that are cooking before they show up, and try to figure out the signs of things that might likely end up becoming threats in different parts of the world. And I think the question you have asked also, tails into that what are the signs that tell us that, okay, of all these things that are the part of the outbreak that is below the surface, what makes some of them pop up to the surface? At times it's as simple as one substitution. At times it could be as robust as a switch or recombination. But the way I look at it is that all those things that are happening below the surface provide the raw material that makes it possible for one or a few to pop up and cross clinical manifestation at any point in time. However, if we can understand the sequence space within which those viruses have room to explore, and we can get a handle on the patterns that's, because if we don't see what is under that surface, we don't even understand the space those things have to explore to be able to, have variants that can cause clinical manifestation. But having the capacity to look at what is happening in that sequence space that they have to explore gives us the capacity to get ahead of these viruses, so that we know that, okay, this is the diversity that exists in this sequence space. Even if this virus is going to sample something, most likely to be sampling within this space, and these are the possibilities that exist. And that can guide us in designing our therapeutics, designing our antiviral agents, our antiviral drugs, so that when that sequence space gets sampled, because we don't know when or why or what will make that happen, but when it happens, we are there ahead of the virus. I think that is what we missed with the current pandemic. And I think that is why we find that it seems to be there's a lot of funding going in the direction of looking for zoonotic agents and sampling animal reservoirs and trying to figure out what is out there, particularly with beta coronaviruses. With respect to clocking, this is the way I understand it. The structural portion of so you can break any virus genome into two parts: the parts that form the capsid or proteins in the envelope, like hemagglutinin or spike protein for envelope viruses, and the portion of the genome that codes for proteins that don't get exposed to antibodies and T cells and the likes, that the immune response does not go after. So because the immune response is going after the capsid proteins, or is targeting them for destruction and flagging them for destruction, they are under heavy pressure to change. It's not as if the virus is consciously changing. It's just that, one, the RNA polymerase is error prone, is making mistakes all the time. And because of that high level of mutation that is going on in that space, the immune response is capturing everything within that space that is not evolving outside of what the immune response is targeted against. And because in any population at any point in time, you have so much diversity, that immune response reduces that diversity and allows the escape, not necessarily that it's doing it intentionally, but it's just that it can capture everything it is targeted against. And those things it can't pick, escape and become immune escape variants and the likes. But you see that kind of pressure does not exist for non-structural proteins because the immune response, the innate immune response or the humoral immune response is not targeting them. There might be some, constraint with respect to protein-protein interactions for those proteins, and if they evolve outside of being able to be involved in those protein-protein interactions within the cell, they might lose their capacity and that virus might not be able to complete its replication cycle. So there are constraints on these non-structural proteins to ensure they can do their function within the cell, interact with the respective proteins they must interact with, but they don't have the constraint to try to escape anything trying to destroy them. That is the way I balance that thought.
Oh, okay. Really, I really agree with now that you shared knowledge. But what actually prompted that question was that the hypervariability lies in the non-structural area, and I think you agree with me. The hypervariability for all this, because you have the intensity of selective pressure at the capsid region, where you have
that's the structural region. So you mean the structural region.
But the non-structural region is where you have this hypervariability. So I'm thinking the hypervariability might be related to why you have a difference in clocking, because these are places where you don't have conservation, and when you say recombination, they are really determinants of how combinations are actually made in species. So I was thinking in that line, but that clarification actually showed me another light that may be the reason why you have another clocking. But now I know better too. So I'm very happy to have you explain that to me. Thank you very much, Dr. Faleye.
Thank you very much, sir. But just to be clear, in case I said it wrongly, if and when I mentioned hypervariable regions, I was referring to capsid portion of the genome, the capsid encoding region, because usually, at least for the viruses I work with, your hypervariable regions are usually in structural proteins, that is in the region that encode the capsid. If I said that wrongly, please, I'm trying to correct myself. Thank you. Are there any other questions, please?
Oh, sorry. Okay, I get you right.
Thank you very much. So is there another question? So while we are waiting for one more question, I think we can still take one more. I would also like to ask, sir, that so what are the challenges that you faced so far in the course of wastewater epidemiology? And I'm coming from the perspective of say an undergraduate student that wants to go into something like that. So what are the challenges one should envisage ahead? Thank you.
Thank you very much for that beautiful question. Well, I wouldn't interpret it as challenges that are particularly associated with wastewater-based epidemiology. In fact, I feel wastewater-based epidemiology is, in a way, I wouldn't say easier, but because I think you have less stringent control on taking wastewater compared to getting samples from people. I think the challenge might be finding the right crew, the right group of people that can mentor you. I think mentorship is the main thing, and I think I've been super lucky finding mentors. I've really been super lucky, super privileged to have excellent mentors around me at almost every time, and I know that has definitely really helped me. So with respect to I think if you are lucky enough to have good mentors around you and you are hardworking and you know for a fact that it doesn't matter what area of research you go into, there will be difficulties. You have to be dogged. Allow me to use that word, because it's usually not a straight line. It is almost never a straight line. You want to go from point A to point D, but the line takes you through point C and point X and point Y and point, and at times you will feel as if you really don't know what you are doing. You at some point you will doubt yourself. At some point and I think it's a good place to be at times, because when you get into a space where your data is not the data you are generating is not consistent with what exists in literature, it is either of two things. It is either you are doing you're doing a really messed up job of everything, or you're stepping into brand new space that nobody has really stepped into before. So it takes a lot of courage, and it's always good, like I said, to have excellent mentors around you to keep your mind sane, because mental health is something, it's a real thing. It's good to have excellent mentors around you that help keep you sane, help encourage you and show you that, okay, help you think through your processes and point you in the right direction. But if there's no substitute to hard work, and putting yourself, as one elder once told me that, and I think the prof that talked this morning also said that it's like any business. What you put into it is what you get out of it. Thank you very much.
All right. Thank you very much. I think you've done good justice to the question. So we so much appreciate having you around on the call. I have personally learned a lot, and I believe everyone else on the call have also. So we'll be wrapping up this session now, and the webinar continues tomorrow by 9 a.m. We'll be having Professor John Heredale, and we'll wrap up the entire series by 10 a.m. Tomorrow also. So please join us again tomorrow for another webinar session. Thank you very much, Dr. Faleye Temitope. Thank you everyone for joining. See you tomorrow.
Thank you for the opportunity. I appreciate it. Thank you very much.
Yes, sir.